Good morning, everyone. In this talk, we will continue to discuss about the particle size, and more specifically, we will be looking at things that you should pay attention to during the development and validation of a particle sizing method. And this can be a method that is used for product batch release to ensure quality, or it can be a method that is used for supporting a bioequivalence determination. Now, through this talk, the goal is that we're able to achieve these following four things together. First, why is it important to determine the particle size distribution, or PSD? Second, what are the challenges? And what are the parameters that are important to consider while you are validating the method? And lastly, what are the common mistakes to avoid, which we will use some example deficiencies to help you understand better. First off, why is it important that you need to develop the right method for particle size measurement? And why is it even important that we want to know the particle size in the first place? Well, we certainly know that for many drug products, particularly those in a dispersed state, such as emulsions, suspensions, liposomes, inhalation powders, or iron colloids. We know particle size is an important quality attribute, and this means that it is critical for the performance of the product, which ensures that the product is not only efficacious but also safe. And in addition, as a generic drug product developer, you might also recognize that particle size is a common attribute that is important for the demonstration of bioequivalence. And here on the right-hand side of the slides are just two examples where in product-specific guidances, the particle size is included as part of the in vitro option to demonstrate BE. And the bottom line is, yes, we know it is important for the measuring the particle size. Again, the question is why? Now, the answer may sound a little bit confusing at first. The particle size is important because what we are really interested in is not the size. If you think carefully, when we analyze the particle size, it is generally not the objective in itself that we are measuring, but rather measuring size is a means to an end. And that end goal can be the physical stability of a suspension product, for example, whether it will settle down very fast or whether the suspension particles will agglomerate. And also, it can be that we're interested to know how fast particles dissolve to be able to enhance the bioavailability. Now, the end can also be to achieve process capability. For example, if the content uniformity of a tablet is going to be impacted by powder flow behavior or not. Or in a very simple way, the end goal can be simply to determine if the two products are the same or if they are different. And I hope you see the point about this discussion about why it's important for us to measure the particle size. It is a purpose that truly matters. And oftentimes we're not interested in the number itself. One example is to illustrate this point. If we look at two products, product A and product B, and I can tell you that the product A is smaller than product B by about 10 nanometer. And I can assure you that the difference is statistically significant. Now the question is, are they really different? And I hope you are not jumping to conclusion right now. And I hope your answer is, it depends. It depends on why particle size is important. Now, let's look at the methods that are available for you to choose. There are indeed many. They vary by the size range that they cover. For example, if it is less than 1 micron or if it is above. And they also vary by the measurement principles. For example, if it is based on Brownian motion of submicron particles, or it is based on the scattering pattern of the particles. And because of these differences, each method has its own unique strengths and weaknesses, and especially when the method is applied to the measurement of a particular particle system. One of the things our FDA lab does is to look at these methods to see what the challenges are and how we should understand them better, and what do we need to pay attention to during the product assessment. And of all the methods that we have explored so far, we know two methods account for majority, indeed about 80% of the usage in submissions. And they are dynamic light scattering, DLS, and laser diffraction. So in the following slides, I will focus my discussion on these two techniques. What are the considerations when you are developing a particle sizing method? First, you should determine if the size range of the technique is appropriate or not. For example, if your product is an emulsion and designed to have a globule size about less than 100 nanometer, then DLS may be more appropriate. On the other hand, if you are measuring a suspension product of about 5 micron, then laser diffraction would have been a better choice. So first thing first, select the more appropriate technique. Second thing to consider is if you are interested in finding out the shape of the particles, or if you know morphology is important. In this case, you might want to use microscopy-based methods such as cryo-TEM as a complementary technique. Now, if you know the particles can dissolve under dilution or aggregate, in other words, the system is sensitive to dilution, now you really need to be careful in selecting the right dispersion medium. For example, making sure you pre-saturate the medium with the drug. And you also need to be careful about selecting the appropriate dilution where it has less impact. In some cases, the particles may not wet very well, so you may need to add some wetting agents such as surfactant, or if they agglomerate during the measurement, in which case moderate in situ sonication may be necessary. And you need to pay also close attention to particle systems that have colors, as this is commonly indicate the particles absorb light, so it may reduce the signal quality in both DLS and the laser diffraction. And some of the settings in the software may compensate for this effect. However, some may require additional effort, such as using more optically transparent cuvette to increase the scattering signal, or just to dilute the sample enough to reduce the light absorption. And sample preparation is another extremely important factor. You need to make sure that the samples are stable and well dispersed. Again, use sonication, dilution, or filtration as necessary, but please make sure you document the sample preparation in detail in your submission so that our assessors can evaluate them. And in addition, we sometimes observe some of the interferences from the formulation components that may require some additional work to develop a good method. And for this, I will provide an example next. In this example, let's look at how excipient may interfere with the size analysis. Here we have a suspension formulation, and it contains a viscosity enhancer, carbomer. And we confirmed using known size standards from NIST that carbomer contribute to laser diffraction signal, which lead to the appearance of a peak right around 15 micron. And one thing to note is that this peak highlighted in red is not real size of carbomer. It looks real, but it's not. So the problem is in the actual drug product, the drug particle is also about 5 to 8 micron, and this resulted in an appearance of a shoulder peak, as this may create variations in the size results and make it difficult to conduct product comparison. In this case, what we did was we developed an interesting method where we use the placebo formulation, which contains everything except the drug, and we collected signal from the placebo as the background immediately prior to collect signal from the actual formulation. And the equivalent software performed the subtraction, and the end result is a clean distribution of the drug substance instead of including the interference. If we go to the instrument setting level, there are other things that you need to pay attention to. For example, laser power, measurement position in DLS, and stirring speed, sonication setting in laser diffraction. And for both DLS and laser diffraction, the optical properties of the particles and the dispersion medium, as well as the analysis algorithm. Also important are the reporting. Is it mean or medium? If it is mean, what type of mean? Volume or surface weighted? Or is it z-average? For DLS, are you reporting cumulative result or distribution result? And we actually will take a closer look at one of the deficiency questions later in this talk. Again, document everything that has to do how data is generated, because those are important for the assessors to evaluate. And last but not least, is your method sensitive to formulation or process changes? For example, if you use a method for purpose of quality control, and you need to make sure that the method is sensitive. And generally, a single technique may be sufficient for either quality control or to demonstrate by equivalence. However, it is sometimes advantageous to try a few complementary techniques to improve your understanding of the product. Next, let's talk about method validation. We know it is important to validate the method to make sure it is suitable for its intended purpose. However, there are some misunderstandings about what is and what is not relevant for particle size validation. In this table below, I listed all the parameters you may find in ICHQ 2R1 guideline, which talks about, in general, analytical method validation. As you can see, some of the parameters are not relevant for size validation. For example, specificity, linearity, range, detection limit, and quantitation limit. Indeed, the most important parameters to validate are the precision and robustness, which I will explain more in the coming slide. One thing that may be relevant for size validation is accuracy, however, not in the way that you may think about it. If you are using, for example, a NIST size standard to show closeness to the known value, what you are actually showing or demonstrating is the instrument or system qualification. Accuracy is always referring to the unknown sample, in this case your actual sample, and how true is your measurement to reflect its size. So we know this is extremely challenging for measurement in a drug product, so you shouldn't view that as your target for method validation. So long story short, it is okay to use NIST size standard in your validation study, but understand that what it provides is the qualification of your system, not necessarily the accuracy. What is really important for size validation are the 3 R's, repeatability, reproducibility, and robustness. So I borrowed this slide from Alan Raw, and many of you may know him. So the first R really measures the machine's performance or the suitability of the method or the sample stability. Why? Because it examines the closeness of multiple measurements of the same aliquot by the same operator in the same instrument and under identical conditions. Indeed, the only thing that is changing in a repeatability study is time. For example, 6 consecutive measurements of the same aliquot in the instrument. And the second R is reproducibility. Now you need to expand to include a different operator and maybe 2 similar but not identical instruments and prepare the sample multiple times. In this case, what you are looking for is if there's any potential issue with sampling or dispersion procedures and if there's any machine-to-machine variability. And the last R is robustness, where you intentionally introduce variations like pump flow rate or sample dilution to make sure you can both detect the changes and also to show it's precise. And these 3 R's are the most important thing in your particle size method validation. Now, let's look at some deficiency examples. First, how should you report the data? In this example, we found the applicant used the dynamic line scattering method and set the acceptance limit of the particle size based on D10, D50, and D90 values. However, it is unclear what is the weight. For example, if it is intensity weighted, which is more closer to the original data that is generated from the instrument, or if it is volume weighted, which would require a transformation using mean theory. If it is the latter, the risk to improper use of the method certainly increases. And furthermore, it is unclear if the applicant applied any special algorithm to the analysis. For example, we know in DLS, some vendor software include general purpose and multiple narrow mode, which may produce very different sizing results. And without this information, it makes it difficult to assess if the method is suitable for its intended purpose. So you really need to make sure you provide the right information in your submission. In the second example, let's look at what is useful to validate. And this applicant validated the PSD method in terms of accuracy and precision for Z average. Again, this is a dynamic line scattering method. However, the drug product is an emulsion, and it becomes important to ensure dilution does not change the size of the emulsions after dilution. In this case, as part of the method validation for the robustness, the applicant should have performed serial dilution to include the range beyond just the target condition to demonstrate that the method is repeatable. And again, what is important to validate are the 3 R's. To repeat, those are repeatability, reproducibility, and robustness. In the last example, we will look at how to set the acceptance limit. One way to do that is based on stability testing results of both ILD and the test product, and more specifically based on the variability observed during the stability testing. And limit should not be set such that to compensate for a stability issue. For example, a particle aggregation over time leading to particle size increase. Instead, root cause of the stability issue should be identified and eliminated. So the acceptance limit is not a solution for stability problem. And we also see very common for the generic applicant to use the batch-to-batch variability of the ILD product to establish the acceptance limit, which is totally fine. However, sometimes we see that the limit was said to be really unreasonably wide, that it almost loses its value in terms of quality and performance. So please set the limit based on the ILD product, but make sure you provide adequate justifications. Here are some more common mistakes to avoid. For example, incorrect choice of the instrument for the measurement, or incorrect use of material or dispersion refractive index. And this sometimes become very important, particularly in a DLS method, if the distribution results are reported. Also, as shown earlier, for example, sometimes it's not clear which analysis was used, whether it's accumulant or distribution. And in general, it is recommended that you use intensity-weighted results if necessary, and avoid the use of volume-weighted values, and certainly do not report in number-weighted results. In terms of the validation, a common mistake is validate entirely based on a reference standard, such as NIST standard. So please make sure you validate the method using the actual drug product. You can use the ILD product in your validation, which is completely okay. And last but not least, make sure you include as much as possible of the details and of your method and your validation, and provide sufficient justifications for critical parameters or settings. And indeed, the more detailed, the better. Now, this concludes my talk, and I hope you are convinced that particle size is an important quality attribute, which is also important for the BE determination. And we need to recognize the strengths and limitation of each sizing method, and we certainly welcome new methods that can provide better understanding. And particle size distribution method should be properly developed and validated, and complete information is important for the correct interpretation of the results and for the assessment. With that, I would like to thank you for your attention and welcome any questions during the Q&A session. Here is a challenge question. What parameters are needed to be critically examined as part of the method validation for PSD? A. Limit of detection and limit of quantitation. B. Accuracy and precision. C. Precision and robustness. And D. Specificity and accuracy.